All right, so a lot of people are asking me what they need to do for today. So the first thing you guys will need to do is you will need to download the Explain Expression of Genetic Information. You can find that here under the attachments for EDay 4. So you download that, you put that into Notability. Copy to Notability. Once it's in Notability, I'm going to create a new note. I'm going to import it all. So there we have my assignment. Okay, so in my assignment here, you will need to, let me change the highlighter to make it a nice color. You will need to obviously put your name. You know my teacher. You should read the introduction, so you need to read that. You'll need to the process and procedures, and then you'll need to do the reading. I've attached the reading in the Schoology folder, so you can easily read about that. Then there's also an additional video that gives you extra information about that. So number 2, it says discuss the following questions with your partner, contribute to a class discussion. You do not need to do that. There is no discussion. There is no class partner. All right, so next step, you will answer these questions, however. What was the problem with hemoglobin proteins that changes in the red blood cells? What problems in the red blood cells causes changes in an organ system? What are the medical symptoms that a person with sickle cell disease might experience? How do the changes in red blood cells relate to the symptoms experiment? All right, so here we are going to fill out the chart, but you're not going to work in teams of 2. All right, and then what you're going to do is there is a mistake here, and that says poster right here and poster right here. That is the chart or the table. That is the chart or the table, okay? And in that chart or the table, you only need to fill out these steps on that table. All right, then number 5, you're going to compare the 2 DNA sequences. The 2 DNA sequences are this sequence and this sequence. Yesterday, you guys learned about different types of mutations, and you're going to figure out what type of mutation there is because there's a mutation in here. So compare the strands. If it's the same length, then it's a substitution mutation. Find where that substitution mutation is. If it's shorter, it's a deletion mutation, and if it is longer, it is an insertion mutation. So read through there and look for the type of mutation that is there, and you're going to answer this question right here. Okay? Then this chart right here, you do not need to do this box, this box. You don't need to do those today. You should, though, fill out this. Why do people... So this is the normal shape, and this is the sick shape. So this is the normal behavior of hemoglobin. This is the normal behavior of hemoglobin. This is the sick behavior of hemoglobin. So you're going to figure out, based on the readings and the videos, what does normal hemoglobin look like? What does sick hemoglobin look like? What does normal hemoglobin act like? What does sick hemoglobin act like? All right, same thing here. You're going to shape of the red blood cells, shape of the red blood cells. What does it look like in a normal person? What does it look like in a sick person? And then what is the consequence of that? So people with sickle cell disease, they are sick. Why are they sick? What are the symptoms? What are the symptoms of having sickle cell anemia? So what are those symptoms? And then this here, you can actually just write here, just normal. There's no, this you can actually write normal in that pace because there's nothing different about somebody that does not have sickle cell disease. I hope this helps you all. I'll be posting this and I'll link this onto the help. All right, bye, Pirates.